in 2019, so we're still a relatively new lab, all things considered. We are interested in understanding the neural basis of social behavior, and more specifically social motivation. So why do some animals find interacting with another animal to be socially rewarding and reinforcing, and why do others find it to be aversive? My personal interests are more selective to aggression motivation. Why do some animals like to fight? Why do other animals find it equally aversive? And within that context, we are also especially interested in trying to understand how this idea of maladaptive social motivation intersects with neuropsychiatric disorders, substance abuse, depression, autism, PTSD, and if there are any new therapeutic approaches that we can try to identify or supplement by looking at the social component rather than other symptoms or signs of the disease. I find aggression to be a really fantastic platform for investigating the mechanisms driving these types of social interactions because it's just so evolutionarily conserved. Typically, the way a lot of aggression research, or just social research in general, has been done is with very passive, exploratory-based behaviors. So what do I mean by that? All right, we have an animal, we put them in an open field, we have another animal, we call it an intruder, we put it in with the resident, and we see what they do. Maybe they groom each other, maybe they walk around and sniff each other, maybe they fight with each other, whatever the reason is. The problem with that is it's completely involuntary, right? There's a lot of difference between going to meet your friends at a movie versus being dragged to a movie you don't want to go to and seeing people there. And obviously, when we look at neural mechanisms guiding social behavior, that voluntary or choice-based component to engaging or not engaging in it is really important, and it's something that has not been captured in the majority of previous work. So the approach that we've taken is moving to what's called operant behavior. It's where animals can lever press or nose poke or perform some operant action to receive voluntarily a social target. So this has really changed things up, because now no longer do we just force animals together and see what happens, but we give the animals a choice of how they want to initiate and maintain or seek out these social interactions. And because it's completely voluntary, now we can tap into a lot of the reward circuitry and motivation circuitry that previously we only looked at in the context of threat and defense. But now, in terms of social behavior, we can look at it in terms of affiliative or reward. And so that's been really exciting. So that's allowed us now to push these things into a more relevant and translational direction. Because when we're talking about human behavior, right, we're talking about that transition from adaptive to maladaptive. This is very seldom because someone is breaking into your house and you want to be alone. I mean, that's an interesting case, but that's not normal. What's normal is I have to choose to go to class or choose not to go to class. I'm not feeling like I want to go to class, but I'm going to go anyway. That's a very, very different set of parameters. And so that's what these new operant models allow us to better model with the animals. And so hopefully, our hope is that these approaches will give us a better understanding of the neural mechanisms that are translationally relevant to the human condition. If we think about social behavior, one of the reasons it's so complicated is because it takes two to tango, right? So, I'm talking about social stress, talking about an animal that's has been stressed out by another animal. Well, that, by definition, depends on the behavior that the other animal is exhibiting towards it. So, we've been using machine learning now in two different ways, both with what we call either supervised or unsupervised machine learning approaches. Using supervised machine learning approaches, we've developed a pipeline called SIMBA, stands for Simple Behavioral Analysis. That's a funny, accidental sort of direction for the lab, which we're now very happy with. It's called Simple Behavioral Analysis, not because the behavior is simple, but we're trying to sort of simplify it for the behavioral neuroscientist who's using it. So what SIMBA has really tried to do is make this supervised machine learning approach very accessible to behavioral neuroscientists. And for this, it's simply for the automation of behavioral classification, whether it's in a single animal, like grooming or freezing behavior, which is very easy, or whether it's in two animals that are rapidly interacting. It's a little more complicated then, obviously. But what that allows us to do is classify large volumes of video very rapidly, and then align those classifications with modern neuroscience techniques like fibrophotometry, optogenetics, neural recording, across the whole, every single frame of video, rather than just what a human annotated previously. You know, I had a very, maybe atypical route to getting here. I started doing research when I was 14 years old. But when I was 14, I joined something called a science research program. At the time, they were very new. Now they're quite common, thankfully, where high school students have an opportunity to get some experience in the lab. You know, this is right like pre-email. So I sent out about 100 handwritten letters to various labs in the area. I got one response, a single response, which I was very grateful for, and I went and worked in their lab, starting as a sophomore in high school, and I stayed there right up until I went to college. And that exposure identified very early on that I really liked research. It wasn't neuroscience, it was just general research. Those early experiences, because I did it from when I was high school and college, I guess have influenced the fact that I think it's really important, very important, to provide access to the lab at an early stage. I mean, when you're a freshman, when you're a sophomore, hopefully before you even decide to be a neuro major here at UW. And I've tried to follow through with that because we have at least 2 or 3 summer high school students every summer. And we have, I think right now, between 8 and 12 undergraduates, depending on when people are, you know, graduating and cycling in. And my hope is that those experiences in the lab provide people the opportunity to figure out what they want to do and if this is what they want to commit to. I also think that we can do better than we're doing. We're doing really well. We can do better. Right now at UW, for example, there are several high school partnerships that we've established. One with the Rainier Scholars, which is a Seattle-based program for middle school and high school students to come and identify research environments and spend a summer or two. And so that's been exceptionally productive, I think, for all of us. We also work with the UW Indoor program quite regularly, which is another program that's specifically for community college undergraduates that are not at an R1 research institution where they'll have access to working in labs like we have here. And then we can bring them for the summer and give them that exposure and then continue to work with them in subsequent years as well. But why is it so important? Well, we have a real issue right now in not just the sciences, but throughout our culture, where there are still systemic barriers to entry. And not just to entry, like we're getting better at that, but really to sustainability and advancement. That's the crux of the issue. One of those I can influence immediately, which is providing access to groups that normally don't have access. And you know, my hope is that by pursuing those approaches, we'll have an effect eventually on the other, which is getting enough people into the pipeline and with support to survive the pipeline, to become professors, to become deans, and provide the perspective that we need, right? That we need if we're going to really have an inclusive and effective environment, training environment for everyone. This is a light sheet microscope. It's one of 2 that we have in the lab. So with light sheet microscopy, what we're able to do is actually image a whole brain in an intact space. So why don't we come take a look in here. So you can actually go on in there. If you go from an angle, a higher angle, you'll see an actual brain. There you go. So you can see, careful, that's close enough. You can see that there's a brain in there, and we have a light sheet on the left and a light sheet on the right, which are sequentially illuminating from either side. With this particular light sheet, what we actually do is end up taking a composite of 2 images, one using the left sheet, one using the right sheet, which then gives us this nice recreation, which gives us the whole intact brain. And then we go all the way from the top of the brain, all the way through the bottom of the brain. Now we've loaded the brain so that we can look at one in a little bit more detail. So we'll go all the way from the top of the brain down. So right now we're on the ventral surface, the dorsal surface of the brain, so the very top of it. And we're slowly going down. Each one of these is a neuron in the brain, and we're going down. And you can see how we can image the whole thing all at once and then go all the way through it. So this allows us to look at the brain in a completely unbiased manner. I'm going to zoom in right now on some of the really cool looking nuclei just to give you an idea. So for example, let's look at some hippocampal neurons. So check this out. So this gives you an idea of the resolution that this technique affords, right? So here we are. We are now going to look at a hippocampus, which is this area right here. And we can slowly zoom in and out of it a little bit so you can see the neurons. Pretty cool. So if we want to go look, for example, at some cortical areas, let's go over here. We can look at the motor cortex, or the sensory cortex, which we can see right here. Look at those neurons, beautiful cortical neurons, right? We can see layer 1 through 6 very easily. You can also see some nice pyramidal layer 5 neurons right there. We can see some layer 6 neurons. We can see some layer 1 neurons and 2 neurons. It's really, really neat stuff.